Okay, so we keep this whole series as anatomy of questions, and here we want to discuss about the detailed structure of a question, because if you understand the structure of a question very clearly, you can answer them fast. Okay, and sometimes understanding the structure is more important, even important than knowledge of the subject itself. So today we will talk about unit 1 and unit 2 questions. Okay, so 6 questions from each unit. Approximate number of questions you can expect from these units in part C in CSIR net exam. So this is okay. So we are back with another session of this anatomy of a question, and this is regarding the last unit for CSIR net exam, that is unit 13. Although last, but it's very important units. 5, 6, 7, 8, even 10 questions sometime can be asked from this unit alone. So give a lot of weightage to unit number 13, that is biological techniques. And why not? Because as a researcher in the future times, when you qualify CSIR net, you need to handle different instruments in the lab. So they ask question from those different instruments, how the instruments work, working principle, as well as most important thing is the function of those different techniques, and exactly when you are going to use those techniques based on the experimental design scenarios. So let's solve a few of this question from unit 13. So this type of the question in unit 13 are mostly analytical. So you need to engage your analytical brain to analyze the data set provided, generally big questions, large questions with so many pictures and graph you need to interpret, and then you can get to answer the question. For example, this one, a 6.4 kb plasmid DNA has two restriction endonuclease sites, HindD3 and EcoR1. Complete double digestion of the plasmid with both enzymes yields two fragments of 3.1 and 3.3 kb. In order to study DNA repair process, a GT mismatch was introduced in one strand of the HindD3 site, and the damaged plasmid was incubated in a reconstituted repair system containing all the factors and enzymes required for the repair. If the efficiency of the repair system is 50%, which one of the following band patterns on agarose gel will be obtained after treating the repaired plasmid with the HindD3 and EcoR1? Whoa, so it's a large questions, and you know the complex sentences are used, and you can see 2 lines, 3 lines with one sentence to make it more difficult. So you know sometimes the problem is to understand the question when you read it. When somebody can explain the question in fragments, you may answer this question fast, but this is the problem that they will check. Generally, if you see this kind of question and if you are already answered 18 to 20 questions already in the part C, you should leave this kind of questions, because this will take time to solve. But you need to understand one thing is that there is this 6.4 kb DNA, and this 6.4 kb DNA is treated with this two different restriction endonuclease, HindD3 and EcoR1, and they generate two different fragments, 3.1 kb fragments and 3.3 kb fragments. Now the problem here is there is a mismatch, and due to the mismatch, now the mismatch need to be fixed by all the DNA repair mechanisms, but they can only fix 50% of the mismatch. So even after fixing the mismatch, we know that even after fixing the mismatch, there will be some portion of the DNA will remain problematic, where the GT mismatch will be present. Now the thing is, if the DNA is with the defect, DNA with the defect means DNA will not provide the 3.1 or 3.3, because this is the normal situation. If the DNA is normal, then only the 3.1, 3.3 fragments are produced, but DNA is defective. So in that case, due to this mutation, the DNA will only provide 6.4 kb fragment predominantly. But if the DNA is normal, then they will produce more 3.1 and 3.3 kb fragments. Okay, so when we see that the 50% of the mechanism, the repair mechanisms used can only fix the 50% of the DNA. So that means what we will see furthermore after the treatment, we will see 3.1 and 3.3 kb bands we will see, but 50% of that, and rest 50% we will find 6.4 kb. So in the data we must find 6.4 kb, 50% of that, and rest 50% equally distributed to 3.1 kb and 3.3 kb. So we need to find out a agar-agar gel picture that explain this. So maximum will be 6.4. So the band will be maximum broad for 6.4 and a thinner for same thinner for 3.1 and 3.3. So the only picture explains this is picture number 4. So this should be our answer. Got it? If you don't understand this, repeat this couple of times so that you can get it. If you read the question couple of times, it will be very clear. Let's move to the next one. Combination of molecular markers with their classification based on either dominant or co-dominant types are shown below. Now this is a question that requires your memory skills and knowing the differences between all the type of markers that we use, that is RFLP, AFLP, SNP, SSR, okay, ISSR and all. So these are few of them. So the question asked is from whether these markers are codominant or dominant, okay, from this one property of these markers. What we mean by this dominant or codominant. The codominant markers follow Mendelian inheritance pattern. That means in the codominant markers can clearly discriminate between the homozygous individual and heterozygous like genotype. It can discriminate between the two, but a dominant marker never discriminate between homozygous and heterozygous. It's going to show only the dominant character, only the dominant genotype there. Okay, so a person with homozygous dominant will show the marker in the same place. A person with heterozygous genotype will also show the marker in the same place. So if we use a dominant marker to find to distinguish between two individuals, one is homozygous dominant and one is heterozygous, we cannot do that. But if we use a codominant marker, we can distinguish between a person who is homozygous dominant for it, homozygous recessive for it, and a heterozygous for it. Okay, so for homozygous dominant there is a specific region to bind, for homozygous recessive specific region to bind, for heterozygous there are two separate places where the marker can be found. Okay, so this is the beauty. So now if I show you the different markers and their dominant nature, RFLP is a codominant marker. STR, SSR is codominant markers, where RAPD are dominant marker. Okay, although the genomic abundance for RFLP and RAPD is high, but SSR is medium. Okay, so now knowing this fact only, these are all the properties are listed. You can read this and remember this so that they can ask you a question from this from other part. You can answer it. So if you read this question, it says the SSR and RFLP both are codominant. So A is correct. So if A is correct, then option 2 and 3 are wrong. Then it says that SSR and RAPD so B is not correct. So if B is not correct, then option 4 is the correct one. You don't need to read anything else. Now if you read the option D, AFLP and RAPD dominant. So RAPD and AFLP are dominant markers. Remember, you should remember RALP, RALP. Okay, RAPD, AFLP. Okay, and RFLP and SSR both RFSS. This is codominant. Okay, you just need to remember. There's no other way to recall it. Okay, a small fraction of clear cellular lysate was run on the isoelectric focusing gel IF to purify the particular protein, which showed a number of sharp bands corresponding to different pi value. The protein of interest has pi of 5.2. Therefore, the band corresponding to pi 5.2 was cut, eluted, an appropriate buffer and subjected to SDS page, which showed 3 distinct bands. One of the following inferences cannot be drawn from the above observation. So what is the observation? When lysate is there, we run the isoelectric focusing and we find out that protein to be present at the pi of 5.2. Then we elute it with appropriate buffer and we also carry out the SDS page. We find out 3 distinct bands in the SDS page. So this is all the events that is done. Okay. Now from all the events, what inference we can draw? Several different proteins having same pi may be present at a single band of IF gel. The answer is yes, because generally this question is from the 2D gel electrophoresis. If you know 2D gel electrophoresis, you can answer this, otherwise you leave this question. In 2D gel electrophoresis, we use 2 different properties. One is the separation of proteins based on their isoelectric point or pi value, and second thing, the separation of the proteins based on their molecular mass. Okay, because many proteins may have same pi or same molecular mass, but proteins with same pi as well as the same molecular mass is very rare. So that's why you use two parameters to separate proteins. So several different proteins may have the same pi value in the single IF gel. Yes, that is true. So one is a true statement. We need to find out the false statement. Okay. So SDS page showed three distinct bands which may represent the molecular mass of different proteins. Yes, it's also true. The protein of interest may be composed of 3 subunits. We don't know whether it's 3 subunit, 1 subunit, 5 subunit, maybe, yes. As the IF gel showed a distinct band corresponding to the pi 5.2, which is the pi of the protein of interest. The protein is composed of a single subunit. We cannot derive this. We cannot derive this conclusion from the result. You know, when you find out a band in 5.2 pi or 5.6 pi or any other places like 8 pi or any places, we cannot tell this for sure that the protein is single subunit or multi-subunit. We say that the protein may be 3 subunit, maybe 5 subunit, maybe 10 subunit, doesn't matter. Because we find out 3 bands. From that we can conclude, yes, the protein may have 3 separate subunits. But by looking at the pi value, we cannot tell whether the protein is of a single subunit or multi-subunit. So this inference is cannot be drawn. Okay, it's wrong one. So 4 is the correct answer in our case. Now let's look at this one. The expression of the hypothetical gene was analyzed by northern and western blot hybridization under control and induced condition. The results are summarized below. In northern blot, we have a control. You know what? Why northern blot is carried out? It's carried out for RNA, like the hybrid bind, like the DNA sequence markers bind with the RNA to find out the presence of RNA sequence. Western blot is to find out the protein sequences. So what we are actually shown here is that RNA is present. For normal, many RNAs are found, and for induced, we also find many RNA, even more RNA, because in induced we find many more RNA, fine. But for protein synthesis, for control we don't find any, but for induced we find many proteins. Why so? So in normal situation, for control, for RNA, in control RNA is present but no proteins are made, but in induced RNA was present, proteins are made. Now the question is, expression of the gene can be regulated by what? So this question can be asked directly without even showing this picture. They show this picture to just make you frightened about this question, and if you see these kind of pictures, people just escape. But actually there is no need. It is simply showing that if we have RNA but we are not making proteins, so in the cell, even if we have RNA, they are not always translated into proteins. Why so? That is due to the the expression of a gene are regulated. Regulated by what? That's what they want to find. So in simple terms, they can ask this question like this: the regulation of a gene expression is done by the following things. This is the basic question, but they complex it with this picture. So what are the factors that actually regulate the gene expression? Control of transcriptional initiation, yes. Alternative splicing, yes. Control of translational initiation, yes. Protein stability, yes. All the 4 factors are actually necessary. They are required for we can say that the regulation of a gene expression. So option 4 with all the 4 statements are correct. Okay, not only this, everything, they are important. If the protein is not stable, obviously the expression will not be carried out, because the proteins are needed for the gene expression as well. Let's look at this one. A chromatin immunoprecipitation assay was performed to determine specific transcription factor binding sites on the promoter of a gene. Okay, pull down was done using either IgG or antibodies against c-Myc. A DNA containing c-Myc binding region was used as a control for PCR amplification, which is known as input in this picture. Which one of the following PCR representations of the DNA is correct? Okay, so what we have here is that we have an input. The input carries a c-Myc binding region. Okay, then we have so we have a DNA with the c-Myc binding region. We have a DNA with IgG sequence, we have c-Myc region again. So pull down was done by IgG, or c-Myc antibody. Now the question is, what type of DNA we will find the maximum in the gel? We know c-Myc antibody is going to bind with c-Myc sequence. So for the DNA where we use anti-c-Myc antibody, we will see maximum precipitation. So maximum precipitation must be visualized here. Which of the options shows maximum precipitation? Option 1. And the least precipitation can be shown for IgG, because we also use IgG, but in this case transcription factor binding site on the promoter we are going to find out, and IgG is not a part of the transcription effect here, because the cell growth and cell division we need mostly IgG is also an antibody, but the IgG cannot bind with c-Myc with that affinity like an anti-c-Myc antibody. So IgG will bind with the least affinity, so IgG will pull down less DNA. Okay, so least will be for IgG and medium will be for the input. So maximum with anti-c-Myc, then with this DNA pairing c-Myc binding region. Okay, so we have the antibody c-Myc, we have a DNA that can bind with the c-Myc for the precipitation, so it's also specific binding, so it's the second one, and the least will be the precipitation done by immunoglobulin G or antibody G. Okay, so this is the third one. So maximum for this one, then second one is for the input, third one is for the IgG. Okay, got it? Small molecular weight compounds affect the activity of luciferase differently. The oil water solubility of a various compound is one property important for its effect on luciferase. The straight line in the graph was obtained by plotting the activity data with 50 different compounds. The luciferase activity of two new derivatives of benzene, A and B, are shown now. So A and B are two different derivatives of a benzene, it's already told, and the options are provided whether either is one is phosphate or propyl or amine or methyl components. You need to know that. Now the thing is, the luciferase activity can increase based on the oil to water solubility. Now you can answer this question by simply knowing the property of a molecule. Property of a molecule, whether the molecule is hydrophobic in nature or hydrophilic in nature. And we know that the oil to water solubility, the more this value, that means the more soluble in oil, that means that particular molecule is hydrophobic in nature, while the oil to water solubility, if it's low, then we can call it hydrophilic in nature. So in this graph what we can see, the oil to water ratio, if you look at here, the maximum is this A, where we have a higher, because this value is higher for oil to water ratio, that means A is hydrophobic and B should be hydrophilic. So which among the following combination gives us a benzene derivative? A should be hydrophobic, B should be hydrophilic. So amine is little more hydrophilic compared to methyl group. Methyl group is more hydrophobic than amine. So obviously 2 must be the correct answer there. Okay. So now if you are going to see this, this picture is going to explain many things. Hydroxyl, you can see polar group, methyl non-polar group, carbonyl a polar group again. So methyl is non-polar, or carboxyl is also a charged group, amino is a charged group, phosphate is a charged group, all these are a charged group. So what we can tell is that obviously the methyl group is obviously non-polar or hydrophobic, and amine in that other hand is a charged group, so definitely hydrophilic. So the oil by water partitioning coefficient for amine will be less, so B should be amine and A should be methyl in this case. Okay. A gene from genomic library is screened by using hybridization technique. After hybridizing the probe, usually a stringent washing step is given. The following statements are given to explain the stringent washing step. 4 statements are provided. Stringent washing takes care of removing unincorporated and non-specifically hydrolyzed probe in the molecules? Yes. Now this question is asked from any of those the process of DNA hybridization assay, like the either western northern blot or southern blot techniques. We need to use these washing steps to remove any unbound DNA from there. So obviously the statement A is correct. So if you know about the DNA hybridization assay, and if you know about the steps of the DNA hybridization assay, like the southern blotting steps, then you can answer this question. Otherwise, leave it. So we know A is true. If A is true, then we can easily exclude option 4. B, stringent washing is done in solution having high salt concentration and lower temperature, or the stringent washing is done solution with low salt concentration and high temperature. Now the thing is, the fact is, if we increase the salt concentration, that can destabilize the DNA, but with a proper low salt concentration can stabilize the DNA which is bound with the probe, right? And the fourth thing, the salt present in the washing solution supports the hybrid to stay intact by shielding the interaction of water molecules. Yes, that is true. So if you know D is true, then we can eliminate option 3. Now we have a tie, 50-50 tie between option 1 and 2. Let's read more statement. Here we know that E and F, reading E and F will not be helpful because in neither in option 1 nor in option 2 we have E and F. So you know E and F are definitely false. Stated salt reacts with DNA molecules and allow easy dissociation of hybrid, definitely not wanted. Stability of hybrid is directly proportional to the temperature, definitely not. Now between B and C, one is true and one is false there, okay? So what is true, what is false? What we know from here is that the stringent washing is done using a low salt concentration and a higher temperature. This is needed because if we increase the salt concentration, instead of stabilizing the hybrid DNA, it will destabilize it. So low salt concentration and high temperature is needed for the stringent washing stage. So C is correct. If you know C is correct, B is false, then we know option 2 is correct. Now this question required very thorough and deep understanding of the steps of the DNA hybridization, otherwise you cannot answer that. We know that we need to use low salt concentration at higher temperatures. Remember, low salt concentration at higher temperatures to achieve a perfect hybridization, okay? In a confirmatory test of HIV, one or more viral antigens are detected in the blood of patients. Following are steps to perform the experiment, okay? So confirmatory test for HIV we are finding out. First of all, this is, if you read the statements, transfer of viral antigen to nitrocellulose paper, incubation with the buffer containing antibodies specific for viral antigens, separation of viral antigens by SDS page, detection of bands by enzyme linked secondary antibody. So this is nothing but a process of Western blotting. Western blotting. So if you know about the Western blotting and we know the steps of the Western blotting, you can answer this question. Otherwise, leave it. So it's a very easy question if you know Western blotting and the steps of the Western blotting. So we are trying to find out the protein and viral antigen, which is obviously a protein. So separating protein from the serum can be done first by SDS page. This should be the first thing. C should be the first thing. Separation of viral antigens by SDS page. Now if you look at the options with the C at the start, there is option 3 and 4. So we can eliminate option 1 and 2. Now we are tying between option 3 and 4. Now read it. Again, what will be the separate thing? After SDS page, obviously we need to transfer it to the antigens to the nitrocellulose paper, right? Because after the process we need to separate into the nitrocellulose paper. So A should be the second thing, because afterwards everything will be done on that paper. So we know CA means option 3 is correct. We don't need to read anything else. But for now I read it. After that is done, what will be done? Incubation with the buffer containing antibody specific to the viral antigen or using secondary antibody. We know first we add the viral antigen, so B, then we add the secondary antibody. So after separating in the nitrocellulose membrane, this is the antigen, let's say this is the viral antigen and this is the unwanted antigen. There is a washing step to take care of the unwanted antigen. Then we first add the antibody specific. So specific or primary antibody, first degree antibody. Then we add a secondary antibody which is either linked with an enzyme to give us the color at the end of the process, or is linked with any fluorescence dye that we can measure, and that with active fluorescence measurement we can understand the concentration of the viral antigen load that is present, okay? Now this one, the most important property of any microscope is its resolution, which is easily determined with capital D and can be calculated from the formula capital D equals to 0.61 lambda divided by n sine alpha, where capital D is the minimum distance between the two distinguishable objects. That is known as the resolution. That means, let's say these are 2 points. Okay, we can resolve it. These are 2 points. Your eye still can resolve it. 2 points. Your eye still can resolve it. Still can resolve it. You cannot resolve it now with your eye, right? So this is the least distance between the two that our eye can see as a separate points. So this is known as the resolution. The minimum distance between two distinguishable objects or two distinguishable points. Okay, in this case wavelength A is this case lambda is the wavelength here of the incident light, and is angular aperture, and n is the refractive index. If you look at n in the refractive index in the medium, given below are the several so sine alpha is the angular aperture, n is the refractive index. Given below several suggestions to improve the resolution of a microscope. Now think about it, if the D value is more, the resolution decreases. If D value is less, high resolution. Okay, so what we can find out, so what we need to do, this lambda, which is the wavelength of light, must be less. So if we decrease the lambda, the D value will decrease, so the resolution will increase. So the first statement, decrease the wavelength of incident light, yes, it can increase the resolution. So A is correct. If you know A is correct, then we don't need to read any other option. We know option 1 is the correct one for this question. We don't need to read anything else here. But still, let's read it. Increase the wavelength of incident light, obviously not. Use oil which has a higher refractive index. Use oil because of its lower refractive index. Now the thing is, obviously we need to use oil with a higher refractive index, because the sign this n is the refractive index. So the value of n will increase, and if n increases, that will decrease the D. Decrease of the D means higher resolution. Okay, so that's why two things need to be done: decrease of the wavelength of incident light and using oil which has a higher refractive index. So A and C, and that has the option number 1 with A and C. So that should be the correct answer. So you know, it's not that difficult to answer. If you know the basics of every single concepts that is there in the CSIRNet paper, you can answer this question quite easily. And in fact, I believe with all these classes are going to help you, and you can answer group C questions now like a champion. Okay? Yes. The answer is yes. If you ask yourself this question, that can you qualify CSIRNet Life Science exam within 6-month preparation? Yes. And if you don't need to believe me, just see the results that our students are getting who joined Shomu's Biology coaching for 6-month period of time.
So I'm really thankful for Shomu's Biology, and I would suggest everyone to go for it.
A lot of help and support by Suman sir, always remembered in my mind.
The study materials I got helped me to understanding the topic very well, and the test series helped me to covering up the syllabus in a perfect time.
This is all because of the online test series.
In-depth knowledge what Shomu's Biology gave me. I want to thank Shomu Biology. I want to thank each and every member of Shomu's Biology for their effort, whatever they are putting on us.
And I am very much happy to choose Shomu sir for my CSIRNet JRF journey.
It really leveled up my preparation. I was confident on the day of exam.
The classes helped me a lot to understand my subject from very basics to advanced level.
So I really thanks Shomu sir and also ma'am for always guiding me.
With proper guidance, roadmap, and the help and training with perfect amount of study material and how to utilize them, the resources, everything together can help you qualify that. I am the person who will help you reach to the pinnacle of your success and your journey for the CSIRNet Life Science examination. If you trust me, if you trust my lectures, then join Shomu's Biology online coaching. Registration is going on. The details are there in the description. Fill the form to reach out to us, or simply contact the number given in this video.